Good morning. Today we are going to discuss about the immunosuppressant. Mainly today we are going to concentrate on the calcineurin inhibitor and m-TOR inhibitors. Now coming to the immunosuppressant agent. Now immunosuppressant are the drug which inhibit cellular or humoral or both type of immune response that have a major use in organ transplantation and autoimmune diseases. So, in case of the immunosuppressant, whenever we are discussing about the immunity, in our last regular classes, we have discussed about the cellular and humoral immunity. What are the different types of immunity and their characteristics we have already discussed. Now, what happens whenever we are going for the organ transplantation and in case of the autoimmune diseases, we one of the treatment approaches, or you can say that it should be in case of the organ transplantation, we need to suppress the immunity for the smooth regimen of this organ transplantation. Now, in those cases, the different drugs are used, which is inhibiting cellular or humoral immunity or the both type of the immunity. Those drugs are called as a immunosuppressant drug. Now, immunomodulation. Whenever we are talking about the immunomodulation, what is mean by that? It mean that it may be the drugs may cause the immunosuppression effect, or the drug can increase the immunity of the body. That means immunostimulatory effect. Now, what are the major mechanism involved into the immunomodulation? That is the antigen recognition and phagocytosis, synthesis of antibodies, lymphocyte proliferation and differentiation, modulation of release of mediators due to the immune response, antigen antibody interaction, modulation of this interaction of course, and the modification of the target tissue responses. So these are the general mechanism of immunomodulation. It involves the immunosuppressant and the immunostimulatory drugs, how they are going to produce or alter the immune responses. Now, these are some basic cytokinin which having the important role in our immunity. Now, interleukin 1 IL-1 enhance the activity of NK cells. This is the natural killer cells. Next is attract neutrophil and macrophages. Similarly, next is IL-2 or interleukin 2. Now, in case of the it is induce the proliferation of antigen primed T cell. Next is enhance activity of NK cell. Next is INF gamma, that is interferon gamma, that is enhances activity of macrophage and NK cell, increase expression of major histocompatibility complex molecules, and enhances production of IgG. Next is TNF alpha. This is the cytotoxic effect on the tumor cell, that having cytotoxic effect. Next is induces the cytokinin secretion and inflammatory responses. Now coming to the classification. Now immunosuppressant drugs are majorly classified into the 5 categories. First one is the calcineurin inhibitor, which is also known as the specific T cell inhibitor like cyclosporin and tacrolimus. Next is m-TOR inhibitor, that is the sirolimus and everolimus. Next is anti-proliferative drugs, cytotoxic drugs like azathioprine, methotrexate, cyclophosphamide, chlorambucil. Next is glucocorticoids like prednisolone type of the drug, and different biological agents or antibodies like TNF alpha inhibitor, interleukin receptor antagonist, interleukin 2 receptor antagonist, anti-CD3 antibody, and that is the polyclonal antibody. Now, today's we are mainly going to discuss about the mechanism of the calcineurin inhibitor and m-TOR inhibitor. Next, in case of the this particular drug, if we see about this, is a complex structure. Clear? This is a complex structure. So here we are going to discuss about the mainly two category of the drug, that is the calcineurin inhibitor and that is the m-TOR inhibitor. Now m-TOR means it is nothing but the mammalian target of rifampin, that is the abbreviated form of the m-TOR inhibitor. Now this is just I'm going to explain about the what is mean by the this mechanism. Here you can find that in the lipin coat, it is nicely this mechanism was explained. Here you can find that this is the antigen presenting cell, clear? And this is the CD80 and 86. These are the majorly involved, which we have discussed previous classes. Now when this that is antigen presenting cell or APC, there will be the you can say that interaction of the T cell receptor. Of course, whenever there is antigen antibody reaction, it will be there. This will trigger. This will trigger. This is the first signal. So due to this APC and the T cell receptor, they will trigger the activation of the calcineurin, a calcium dependent phosphatase. Now what will happen? This calcium dependent calcineurin, it will get activated due to this particular signal. Now what is the role of this calcineurin? Now if you see about this calcineurin, this is already this is you can find that this is the nucleus, so this whole portion inside that nucleus, and this is the cytoplasm. Now that is the NFATC, that means cytosolic nuclear factor of activated T cell, that is present into the phosphorylated form, and you can say that when there will be the activation of the calcineurin, that is the inactive NFATC to active NFATC. Now this active NFATC, that means cytosolic nuclear factor of activated T cell, it will enter into the nucleus, and this, if we see about this, you can say that active NFATC, they are going to have the effect on the IL-2 genes. So here, as we have shown that, say first what is happening again, if we are discussing activation of the T cell receptor, result increased into the intracellular calcium. So already we have shown here T cell receptor activation, because there is a antigen presenting cell, so there is a increase intracellular calcium. Next is what will happen, activation of the calcineurin. So here the activation of the calcineurin, and as a result inactive to active NFATC. As a result, dephosphorylation by calcineurin already we have discussed. So NFATC moves from the cytoplasm to nucleus. So it is transportation from the cytoplasm to nucleus. Next, what will happen? NFATC associate with the other nuclear components, leading to the activation gene to the encoding cytokinin. Now cytokinin means here mainly we are concentrating into the IL-2 genes. Now when there will be the IL-2 genes, it will be there will be the, of course, if you remember that protein synthesis process. Now in the protein synthesis, what will happen? From this, there is a synthesis of the mRNA, and based upon the codon, there is a presence of formation of the different proteins, specific proteins. So from this mRNA, of course, there is a binding with the ribosome and all, and then there will be production of the IL-2, interleukin 2, clear? The interleukin 2. Now this interleukin 2, now it is present into the cytoplasm, and there will be the release of the IL-2. And already we have discussed that IL-2 having one of the important role into the cell mediated immune response. Already this section we have discussed. So this part, what we are seeing again, if we are doing in the first case, activation of the T cell receptor after combining with the APC. As a result, what will happen? Increase the intracellular calcium. Next is activation of the calcineurin due to the calcium. Next is dephosphorylation of NFATC by calcineurin Now this active NF-ATC, it will move into the nuclear, then it will act on the cytokinin genes, IL-2 genes, and as a result IL-2 mRNA, and there is a formation of IL-2 interleukin 2. Interleukin 2, it will come out from the cell and it will playing an important role in the cell mediated immunity. Now in case of the calcineurin inhibitor, mainly we are going to see about the two type of the drug. One is the cyclosporine and tacrolimus. These are the two drugs, cyclosporine and tacrolimus. Now what will happen? This cyclosporine, it will enter, it will bind with the cyclophilin. Clear? Cyclophilin, this is the protein, so it will bind with the cyclophilin. So there is a complex of the cyclophilin and cyclosporine, and then what will happen? They will inhibit this activation of the calcineurin. Clear? So cyclosporine, it will bind with the cyclophilin and they are inhibiting the activation of the calcineurin. Similarly, if you see about the tacrolimus, it will come but it will bind with the FK binding protein. Here it will bind with the FK binding protein and they are inhibiting the calcineurin. So both the drug, it is inhibiting the calcineurin, but tacrolimus, it will bind with the FK binding proteins, whereas the cyclosporine, it will bind with the cyclophilin. As a result, this you can say that inactivation of the calcineurin. Now inactivation of the calcineurin, or it is inhibiting the activation of the calcineurin. What will happen? So these particular steps, it will be inhibited. Now there is no activation of the NF-ATC. So ultimately, as a result, what will happen? This IL-2, it will not going to be synthesized, and thereby they are inhibiting the immune response, because IL-2 synthesis, it will be inhibited. So this is the mechanism of this cyclosporine and tacrolimus. Now coming to the second class of the drug for today, that is the sirolimus and everolimus. Now they are going to say here what will happen. This IL-2 receptor is there, so IL-2 after synthesis, it will bind to the IL-2 receptor. They will send the signal to the m-TOR. What is m-TOR? This is the mammalian target of rifampin, and then they will cause the cell cycle, causing the cell cycle activation. So what will happen? Mainly sirolimus, it is inhibiting the m-TOR, and the cell cycle activation, it will be inhibited. Now here we are showing the same thing. IL-2 receptor, it is causing the activation of the m-TOR. Now what will happen? m-TOR increase the translation of the selective mRNA that promote the transition from the G1 to GS phase of the cell cycle. So as we have shown here, we have shown here what is the G1 to S phase. G1 to S phase is there. So promoting. Now what will happen? Sirolimus, it type of the drug, it will bind with the FK binding protein. So here also we have shown, in case of the tacrolimus, they are binding with the FK binding protein, and here also we are saying the sirolimus or everolimus, they are binding with the FK binding protein. Now after binding with the FK binding proteins, this sirolimus and FK binding proteins complex inhibit m-TOR. m-TOR, this will be inhibited, thereby inhibiting the translation and causing the T cell to arrest G1 phase, and causing the T cell to arrest the G1 phase. By this way, it is suppressing the immunity. Yes, it is suppressing the immunity of us. So I think you have understood what is the basic mechanism involved into these two category of the drug. Now this is a general picture. Of course, the other drugs we will discuss into the other classes. Now if we are seeing into the mainly antigen, so due to the antigen, T cell receptor, it will be activated, activation of the calcineurin, dephosphorylation of NF-ATC, IL-2 gene promotion, production of the IL-2, it will act on the IL-2 receptor, progression into the cell cycle and cell proliferation. This is the normal pathway how you can say the cell proliferation it is acting. So in today's class, whatever we have discussed, that is the cyclosporin and tacrolimus. So they are inhibiting this one. The block the calcineurin and inhibit the IL-2 synthesis. Already we have discussed. Clear? Already we have discussed. And tacrolimus also block calcineurin and inhibit IL-2 synthesis. But of course their target is different. In case of the cyclosporin, they are going to act on the cyclophilin, and tacrolimus, that is the NFK binding protein. Whereas in case of the sirolimus, as we have told, that is going to block the m-TOR, and that's why it blocks cytokinin stimulated cell proliferation. Cytokinin inhibited cell proliferation because they are binding the m-TOR. So these are the different drugs. Cyclosporin mainly it is used into the organ or bone marrow transplantation, as well as the different inflammatory conditions such as ulcerative colitis, rheumatoid arthritis, and atopic dermatitis. In case of the tacrolimus, it is used to prevent the organ transplantation rejection and to treat moderate to severe atopic dermatitis. Similar, next is sirolimus. This is the prophylaxis for the organ transplant rejection. And adverse effects: cyclosporin, renal dysfunction, tremor, hirsutism, hypertension, hyperlipidemia, gum hypalgia. Tacrolimus, nephrotoxicity, neurotoxicity, GI problem, hypertension, hyperkalemia, hyperglycemia, and diabetes. Sirolimus, increased serum cholesterol and triglyceride level, anemia, leukopenia, thrombocytopenia, or hyperkalemia, fever, GIT different problems, and it may delay the wound healing. So these are the different drugs under these two categories. Thank you.